This week in lab, we'll be completing Exercise 38, Anatomy of the Digestive System, and Exercise 39, Digestive System Processes. The major role of the digestive system is to provide the body with the nutrients, water, and electrolytes essential for health. The organs of the digestive system ingest, digest, and absorb food, and eliminate the undigested remains as feces. For ingested food to become available to the body's cells, it must first be broken down into smaller diffusible molecules in a process called digestion. These digested molecules can then pass through the epithelial cells lining the digestive tract and into the blood for distribution to the body's cells. This process is called absorption. The digestive system consists of a hollow tube extending from the mouth to the anus, into which various accessory organs empty their secretions. In this figure, organs marked with asterisks are accessory organs, while those without asterisks are alimentary canal organs. Note that the spleen is pictured in the figure, but it's not a part of the digestive system. The organs of the digestive system are traditionally separated into two major groups: the alimentary canal, or gastrointestinal GI tract, and the accessory digestive organs. The alimentary canal consists of the mouth, pharynx, esophagus, stomach, small intestine, and large intestine. The accessory structures include teeth, which physically break down the food, and the salivary glands, gallbladder, liver, and pancreas, which secrete their products into the alimentary canal to aid with digestion. The alimentary canal is a long tube that runs from the esophagus to the anus. There are twists, turns, and sphincters along the way. The different parts of the alimentary canal are similar, but the basic plan is modified along the way to provide specific digestive functions. For example, the small intestine contains villi and microvilli that increase surface area, allowing for a greater absorption of nutrients. The alimentary canal has four basic layers, or tunics. From the lumen outward, these are the mucosa, the submucosa, the muscularis externa, and the serosa. Each of these layers has a predominant tissue type and a specific function in the digestive process. The table in your lab book summarizes the characteristics of the layers of the wall of the alimentary canal. Food enters the digestive tract through the oral cavity, or mouth. Food is mixed with saliva and is masticated, or chewed. One component of saliva, salivary amylase, begins the digestion of starchy foods within the oral cavity. Both the mechanical breakdown and chemical breakdown of food begins before the food has even left the oral cavity. After the tongue initiates swallowing, the food passes into the pharynx. The pharynx is divided into three parts: the nasopharynx, oropharynx, and laryngopharynx. The muscles that make up the pharynx initiate wave-like contractions that propel food into the esophagus. The esophagus is approximately 25 centimeters long and extends from the laryngopharynx through the diaphragm to the stomach. The esophagus has no digestive or absorptive function and is essentially a food passageway that uses wave-like motions, called peristalsis, to move food to the stomach. The gastroesophageal sphincter is located at the junction of the esophagus and the stomach and controls food passage into the stomach. The stomach is located in the upper left quadrant of the abdominopelvic cavity. Smooth muscle within the stomach mechanically breaks down food, while gastric glands secrete hydrochloric acid and enzymes to chemically break down food. After food is processed in the stomach, it becomes a creamy mass called chyme. The chyme then enters the small intestine through the pyloric sphincter. The small intestine is a convoluted tube about 6 feet long that extends from the pyloric sphincter to the ileocecal valve. The small intestine has 3 subdivisions: the duodenum, or the shortest section right after the stomach. The jejunum, the second portion, lies mostly within the umbilical region, while the ileum, the third portion, lies mostly within the pubic region. Brush border enzymes, enzymes from the pancreas, and bile all enter the duodenum to complete the digestion process. Nearly all nutrient absorption occurs in the small intestine, where microvilli, villi, and circular folds increase the absorptive surface. The large intestine is about 5 feet long and extends from the ileocecal valve to the anus. The large intestine is divided into several regions: the cecum, with the appendix hanging from it, the ascending colon, the transverse colon, the descending colon, the sigmoid colon, and finally the rectum and anal canal. The major function of the large intestine is to consolidate and propel the unusable fecal matter toward the anus while reclaiming water from the undigested food. Bacteria within the large intestine also manufacture vitamins B and K. The accessory organs of the digestive system are not a part of the alimentary canal, but assist with digestion by secreting and storing important substances such as digestive enzymes and bile, an important lipid emulsifier. 3 pairs of salivary glands empty their secretions into the oral cavity. The liver is the largest gland in the body and performs many metabolic roles. Its digestive function is to produce bile. Excess bile is stored in the gallbladder until it is needed for digestive processes. The pancreas is a gland that has an endocrine function, producing the hormones insulin and glucagon, and an exocrine function, secreting enzymes into the duodenum to neutralize acidic chyme. The food we eat must be processed so that its nutrients can reach the cells of our body. First, food is mechanically broken down. Then, food is chemically digested by enzymes into molecules that can be absorbed. In lab, we will study digestion processes by 3 different enzymes. The first is the digestion of starch into maltose by salivary amylase, an enzyme produced by salivary glands. The second is the digestion of protein into smaller peptides by trypsin, an enzyme produced by the pancreas. Finally, we will study the 2-step emulsification and digestion of lipids into monoglycerides and fatty acids by bile, an emulsifier produced by the liver, and lipase, an enzyme produced by the pancreas. In lab, we will perform a series of tests to determine whether digestive enzymes have successfully broken down carbohydrates, protein, and lipids into their basic molecular units: sugars, peptides, monoglycerides, and fatty acids. Before coming to lab this week, read through exercises 38 and 39 and take your pre-lab Canvas quiz the day before your scheduled lab. As always, reach out to your lab instructor if you have any questions or concerns.